In this video, we are going to look at some points of emphasis about carbohydrates. So let us show you this one. So again, from the word carbohydrates, so carbo is from carbon and hydrates is kind of like hydra or water. So you can think of it as the hydrates of carbon, right? Because if you think about that, the carbohydrates always has a ratio of 1 is to 2 is to 1. And if you just focus on the H and O, H2 and 1O, it's just H2O. So you can think of it as hydrates of carbon, right? So the ratio is 1 is to 2 is to 1. It's the hydrates of carbon, right? Obviously, that is a loose way of describing it, but that is the etymology or the origin of the word why it's termed as carbohydrates, right? So that's the first point of emphasis. Now, so you can have the formula CH2O. Now, carbohydrates have three different categories, so to speak. So the first category is what we call a monosaccharide, and then disaccharide and a polysaccharide. So this saccharide part means sugar, saccharum. So carbohydrates are basically sugar molecules or sugar structures or structures based off of sugar molecules. So you have monosaccharides, disaccharides, and polysaccharides, and as the video explains, one for mono, di for two, and poly for many units of sugar. Right. So the first so that is the thing that you have to know. That's the second point. The third point is that the monomers for all of these are the monosaccharides. So even if we talk about disaccharides and polysaccharides, the monomers, meaning the bases or the basic units, just like in nucleic acids, the basic units are nucleotides. In this case, the basic units for all the carbohydrates are monosaccharides. And you have three kinds: glucose, fructose, and galactose. So glucose is what we use in our bodies, right? So even in photosynthesis, organisms produce glucose, and that glucose is passed on to the secondary consumers when the consumers eat the producers, and then the primary consumers, and then the secondary consumers get the glucose from the bodies also of the primary consumers. So all in all, the source is really the plants because of photosynthesis. And then fructose is the sugar in fruit, and galactose is the main sugar that makes up milk, right? That's why you have the lactose there, but it's called galactose. Now, if you look at the structure of all this sugar, you can have the cyclical form, which is in this form, and then you can have the straight chain form. Now, again, if you go to biochemistry, it's even more complicated than that. You can have the dextrose version and other versions, and but for high school biomolecular study, this is enough that we express that they could be in straight chain form and they could be in cyclical form. Now you can pause this video here and you can count the number of carbons, but you bet it would always be C6, H12, and O6. There will always be 6 carbons there, H, 12 H's, and 6 oxygens. So by the way, these glucose, fructose, and galactose are called hexoses, meaning these are sugars that have have 6 carbons, hexoses. If you remember when we discussed about in the third quarter, when we discussed about the deoxyribose and ribose, those are pentoses. Those are those are sugars with 5 carbons. But these are hexoses. So remember, it's C5, it's 1 is to 2 is to 1. So for a hexose, obviously it would be C6H12O6. If it's pentose, it would be C5H10O5. So such is the formula for your deoxyribose and ribose, right? So that's the idea. So the next point of emphasis that I would like to for you to focus is this idea that you can differentiate glucose and fructose and galactose by the shape first. So let me remove myself because I'm uh, in the way of the illustration. But the idea here is glucose has a hexagonal form, and galactose also has a hexagonal form, right? And fructose is the only one that is a pentagonal form. But take note, pentagonal doesn't mean 5 carbons. It still has 6 carbons, but the shape is pentagonal. And since one is occupied by oxygen, just like the pentose, remember? The pentose, I mean the deoxyribose and ribose, that means only 4 can be accommodated in the inside. So this is 1, this is 2, this is 3, this is 4. One, where's the fifth and the sixth? It's outside. This is the fifth carbon and then the sixth carbon, right? So you would single out fructose just by the shape because it's pentagonal. Now glucose and galactose, it's a little bit harder to um, to point out the difference. But if you look hard enough, you can find that here in this side, the OH is on top, while the H is at the bottom or at the bottom. But in the galactose, H is at the top and OH is at the bottom. So therefore, that's the only difference between galactose and glucose, right? So they are both hexagonal in shape, but OH in glucose is on top, H is at the bottom. For galactose, H is at top, OH is at the bottom, right? But make no mistake. But you might be asking, sir, I don't remember plants producing fructose or plants producing galactose. I usually just study that it's the photosynthesis creates glucose. So how do these things create glucose, fructose, and galactose? Well, as you can see, because they are just same formula C6H12O6, you can bet that if you have glucose, you can convert it into fructose. And if you have fructose, you can convert it to galactose. So you can interconvert between these two. Although it takes more energy in the case of the cells, but you can technically produce fructose and galactose from glucose. So that's why in photosynthesis you only create glucose, but you can interconvert it into fructose and galactose. So that being said, the basic units are still glucose, fructose, and galactose, right? So if you move past it and you go to the disaccharides, you can have glucose and glucose bonded together. So that's the next point of emphasis also, which is that the bond that forms between them is called a glycosidic linkage. So remember, for nucleic acids, it's phosphodiester bond. For the proteins, it's a peptide bond. For carbohydrates, the link or the bond is a glycosidic linkage or a glycosidic bond, right? So glucose and glucose can form a disaccharide, and that's called maltose. Glucose and fructose is sucrose, which is table sugar. This is what we get. And glucose and galactose forms lactose. So again, science class is not straightforward, and I believe that probably what happened here is that people discovered milk, and then they discovered the carbohydrate related to milk, so they called it lactose. And then they realized that lactose is just a higher structure of glucose and another sugar molecule, which is a derivative of glucose, and that's why they call this galactose, right? But obviously, if science was straightforward, I would have called this a simpler name and this a more complicated name, just to emphasize that these are the simple units that make up this more complicated unit. But again, science is not straightforward. Sometimes you discover the higher structure first before the basic structure. Sometimes you do the opposite. And so it's not as straightforward as like that. But the idea is glucose and galactose forms lactose. And look at the bond between maltose, sucrose, and lactose. If you notice, there's a difference in glucose and galactose, right? And that is uh, because of the where the bonds are created. So for example, if you look at this, let's pause this video right here. If you are going to, let's say, bond um, glucose and fructose, you would need the H here and you would need the OH here. So think of it as this bond here where the H is supposed to be, and this bond here where the OH is supposed to be. That's the bond that would connect the glucose and the fructose. Okay? So if you notice, the H here is at the bottom, and the OH here is at the bottom also. So they are both at the bottom. So that's why when you bond them, this is at the bottom, and this is also at the bottom. So they will just bond like that, right? But if you look at galactose and glucose, if you connect galactose and glucose, if you look at OH, H here, glucose is at the bottom. I'm sorry, I'm in the way. H at the bottom. But so you now need an OH to bond it with, right? So if you look at the OH here, on this side, it's at the top, right? So it's at the top. So what happens then is the site available for bonding here is at the top, but the site available, so at the bottom is for the glucose, but the site available for the galactose is at the top. So now when you bond them, you create this bond here. And this bond is what we call a kind of like a trans bond. Trans meaning at opposite sides, right? Opposite sides. And the only reason why I'm discussing this is to emphasize that lactose is kind of different from all the disaccharides because of this bond here. The bond is different. And guess what? This bond, which is a trans bond, is much harder to break than this configuration here. So that's why when you have maltose, malt sugar, basically the one that you can find in malt products like beer, it's easy to break down. For sucrose, table sugar, it's easy to break down, but lactose is difficult to break down. That's why we need a enzyme for that, a lactase. And some people either do not have lactase or they lose the ability to have lactase in their system when they grow up. So people have lactose intolerance, right? So that's my point there. That's why I zoomed in into this concept of glucose and galactose has a different bond than glucose fructose or glucose glucose disaccharide, right? So moving on, we have polysaccharides. Polysaccharides is again a long chain of monosaccharide units, and you could have straight chains or branch chains, and if these straight chains of glucose molecules or branch chains are in plants, we technically call it starch. So let me get this straight. If it's straight chain, it's called amylose, right? If it's branch, it's called amylopectin, but both of them are present in plants and both of them can be called as starch. Both of them can be called a starch. So starch can refer to amylose, starch can also refer to amylopectin, right? But that's plants. How about animals? Animals do store energy also, glucose. So that is glycogen. Okay? And again, let me remind you that when we discussed about the pancreas, when you eat a lot of sweets, you have a lot of glucose in your blood. Again, your pancreas produces insulin, and that insulin would allow the glucose in your blood to actually be stored in your liver and in your muscles. How are they stored? They are chained together in a highly branched configuration called glycogen, and that glycogen is in your liver and your muscles, right? So amylopectin, amylose starch for plants, glycogen for humans and animals. And then you can also have structural molecules. Structural molecules meaning these are molecules we don't use for energy, unlike starch, amylopectin and amylose, we use that for energy for plants. And glycogen, we use that as energy source for animals. But these are structural molecules. These are molecules that we use to create the parts of our body structurally. So parts of our bodies. I mean, when I say our, I mean us, us as an organism, right? So cellulose, chitin, and peptidoglycan. So for example, cellulose. Um, cellulose is just these. So glucose units, but these glucose units are layers, and it's not like it's just one layer and then one branch. This one is layered, and because it's layered, again, it now is very hard to break compared to just the ordinary amylopectin and amylose, right? So to the point that organisms that eat a lot of plant material like cows and cattle, they have evolved to have multi-chambered stomachs, right? If you remember, the difference between cattle and us is that we have one chamber, the stomach, but for the this cattle things, they have different chambers, and they do that so that they can still ferment the cellulose and they can break down. So cellulose is very hard to uh, digest, and well, it serves its purpose because it is used as the barks of the tree, for example, and it's structured, so it has to be firm, right? It must be not that soft. So that's cellulose. And then you can have also chitin. So chitin is, I believe it's pronounced as chitin. It's found in the exoskeletons of crustaceans and insects. And in fact, I know some of the sci-high students actually have, this is actually a favorite kind of investigator project using the chitin that you can extract from these organisms to some applications or other. But it's just really a chain of glucose plus some amino groups. Okay, so if you have a chain of um, sugar molecules, monosaccharides, plus you add some amino groups, that is chitin, and you can use that, and nature uses that as exoskeletons of insects and crustaceans, right? But after amino groups, remember, you can chain amino acids and form a chain of amino acids. So that's the next step. So for bacteria, bacteria uses a chain of uh, monosaccharides, but they have added in peptide chains. Remember, a peptide is a chain of amino acids. So for think of it as, again, so that it's easy to digest. The amylose, amylopectin, and glycogen are just a chain of amino acid, a chain of sugar molecules. But for the chitin, it's a chain of sugar molecules plus some amino groups, right? For peptidoglycan, it's not just amino groups, but peptide chains, meaning a chain of amino acids together. So these circles here represent chains of amino acids or a peptide chain. Again, we don't call it a protein yet because, again, we have emphasized that if it has no secondary or tertiary structure, it's technically called a polypeptide. So these are peptide or polypeptide chains that are incorporated in the chain of monosaccharides. So that's a peptidoglycan, and peptidoglycan are the cell walls of bacteria. And actually, um, this is the reason why alcohol can also kill bacteria, because the alcohol can denature, or the alcohol or alcohol heat and other stuff can denature the amino acids here. Remember, amino acids are just chain, so it can denature the chain of amino acids here, just like you can denature a protein using heat, like cooking meat, for example, and that makes the integrity of the cell wall susceptible, and you can now attack the bacteria and kill it, right? So in summary, you can have simple sugars and complex carbohydrates. And when we say simple sugar, we usually mean the monosaccharides and the disaccharides, and that is already 6 of them, right? Glucose, fructose, galactose, and then for the disaccharide, maltose, sucrose, and lactose. And then for complex carbohydrates, you can have 3 of them, which we use for energy storage, 2 for plants, amylose and amylopectin. We call it starch. And then glycogen for animals, and then for structural, you can have cellulose for plants, and then chitin for insects and crustaceans, and then peptidoglycan for bacteria. Okay, so that is all for this video.